Thank you, and good morning to everybody. So my name is Tom Westerling, and as mentioned, I work at the Dana-Farber as a research fellow. So my presentation will be the testing that I performed in order to make sure that the machine is working and to make the decision that what we are going to start working both at the center and in our lab. So I did basically two tests, so I'm going to show both of them. So one of the tests was just normal histone metal mark chip followed by library construction and sequencing. And the other test is basically one of the most simple chip experiments you can do, ER-alpha, estrogen receptor alpha precipitation from estradiol stimulated MCF7 cells, and comparing that to what used to be my standard method, which was e-gel, so the in-vitrogen e-gel system, which is also a simple but a little bit more complex method than the Pippin prep. So that's basically what I'm going to use my time for. So. And so in this gel, I'm just highlighting kind of what kind of material I started with and what I ended up yielding. So I did microcochlear nucleus digestion on normal cells that were treated with different drugs, and then after that I performed a histone metal chip and took 10 nanogram of this chip and run that after the ligation of the adapters, run them either on Pippin prep or on the e-gel, and followed that up with amplification. And the post-amplification material is what you see on the left-hand side in the gel. So in lane 1, I tried to capture the mononucleosome band, and I obviously missed that in the e-gel. In lane 2, I just as a backup took the dinucleosome. That one I was able to capture, so that one looks great. And in lane 3, I did it the easy way, and that's giving Pippin prep the command to collect between 165 and 210 base pairs. And then after amplification, this is the band that comes up. So this was encouraging, and I continued on doing actual real experiments on the Pippin prep only, and I did single end sequencing on HiSeq 2000, and I got on average 90 million reads, of which between 70 and 80 million were uniquely mapped. And then going on with our downstream analysis, actually this is proved to be a really useful data set, and if somebody knows about nucleome positioning calling when using the NPS software, I was able to call 300,000 position nucleosome with high confidence. And in the next shot, I have just a brief overview. So this is just a BAM file displayed in UCSC genome browser, so you can see the kind of sequences that you're going to get. So these are 3 different treatments of the cell line. And you can't, from this, you can't gain any real biological information. This is just to show the kind of average coverage that you will expect to see over 1,100 kb range. So with that, so the conclusion from this is basically that it's for a narrow size range that you can't really see. You know it's going to be at 180, but you can't see it. The Pippin prep is really useful because you can just give it a broad swatch around that peak region that you're expecting, and then you can go and drink some coffee, and the machine will take care of the rest. So the following step is a more direct comparison head-to-head of taking ER chip material from MCF7 cells. I started with about 3 nanograms of chip DNA, 75 nanograms of input DNA, and then I split the reaction after ligation, and then did my normal e-gel size selection, or then a Pippin prep size selection. And then after that, I proceeded to do amplification, and the cycle amounts I've just listed on the gel, so that's the 12 or 18 cycles. And hopefully you can see, if you go to compare the Pippin prep left-hand side versus on the right-hand side, you have the chip and the input. Oh, sorry, actually I have a little bit of a marking error here. So okay, so let's start from the left-hand side. So I'm doing Pippin prep input 12 cycles. and you can see that the input is amplifying well at 12 cycles because there were more starting material. At 18 cycle, it already starts to have a little bit of a problem from driving the PCR too far. And in the eGel, likewise, it's working fairly well at 12 cycles. You can actually see if your screen is good enough. You can see that I have collected 3 independent sampled 3 times over the while the eGel was running. So that's why you see 3 distinct bands there. That was expected. And on the right-hand side, we had ChIP. I've driven it all the way out to 18 cycles. And again, you can see from the Pippin prep at 18 cycle, you have a fairly clear band. The second marker here is 200, and it goes all the way up to between 400 and 500, which was what I selected for. And you can see that the size selection is working fairly well, and the yield looks pretty good. And it's comparable to what you would get from an eGel, which is also an efficient system. But with the eGel, you don't get this broad size range, and you might miss stuff. So I did not end up sequencing this particular sample. I did do bioanalyzer on it and some qPCR testing. So on the left-hand side, you can again see the Pippin prep, and on the right-hand side column, you see what happened in the eGel. So on the Pippin prep, everything looks as normal, and the bioanalyzer looks very, very clean and nice. I'm using the high-sensitivity dilution of the sample and the high-sensitivity chip. And on the eGel side, this would not have ended up going on a sequencer because I wasn't able to resolve the primer-dimer pair, and even if I would have done that, the enrichment of the material would have been so low that would not have been a good run on the ChIP-seq 2000. And this is also confirmed when I do qPCR with primers to negative and oh, sorry, I'm skipping ahead on a slide. So parallel samples to this one that I did perform on doing both post-ligation size selection and post-PCR size selection. Doing Pippin prep on those, I did not have comparable samples on the eGel. So here is, you can see 2 independent libraries starting from 5 nanograms or 10 nanograms. That's the yellow of 10 nanograms and the red trace of 5 nanograms. And the input is the green, which I started 50 nanograms. And all of the peaks called by max are where they're supposed to. And this is just an example of where you can go down to fairly low amounts. I had 40 million uniquely mapping reads in the 5 nanogram sample. So I'll just flash the last slide. So showing that as usual, when you do qPCR validation after enriching the library, you do see a little bit of higher enrichment of the ready library compared to the original. So the prepared library is the blue, and the original material that is completely unamplified is in green, and then the size select eGel of 2% is the red one. So again, based on this one, I would never have gone on sequencing the eGel, so that was a state library with the Pippin prep, it worked. With that, I think that the conclusion that I have to say is that I think that 90% of the people in our lab at the moment are using the Pippin prep, and nobody is going to go back anywhere to anything else.
Okay, thank you, Tom.